Throughout history, there have been many plagues and viruses, but none have so obviously evolved and strengthened to infect humans as many times as the flu. There have been 3 major outbreaks of the flu, once in 1918, once in 1957, and once in 1968. Each of these outbreaks was caused by a different hemagglutinin subtype, which basically means a different hybrid form of the virus. Hemagglutinin is a glycoprotein found on the surface of RNA. But first off, what is a virus? The flu virus is a microscopic parasite composed of 8 strands of RNA that attach to a host source in order to survive and reproduce. Because it cannot reproduce without a host, a virus isn't considered a living organism. The flu is believed to have originated in 3 mammalian hosts: pigs, birds, and humans. The reason that the flu virus was able to change and hybridize is evolution. There are 3 types of the mammalian H1N1 influenza virus: BM1918, the seasonal flu virus, and the classic swine flu virus. These viruses are made up of 8 genes: PB2, PB1, PA, H1, NP, N1, M, and NS. During the 1918 pandemic, there was varying severity to the people infected by the virus. This is thought to be explained by the co-circulation of both BM1918 and the seasonal flu virus during the pandemic. Components of the H1N1 virus circulated in people and piggies 2 to 15 years before the horrible pandemic. In 1889, the gene diverged to become the precursor for mammalian H1N1, but it wasn't until 1904 that the gene diverged to join the other components of classic swine and BM1918. In 1914, the gene diverged to also help create the seasonal flu. The most recent common ancestor for the H1 gene is around 1885. From then, it became classical swine on one end and the precursors for BM1918 and seasonal on the other. Around 1905, the H1 involved in the BM1918 virus began circulating, and the H1 involved in the seasonal flu virus started circulating in 1913. Mammalian H1N1 genes all formed a monophyletic group, but only 2 of the genes, PB1 and PA, have a small avian clade that is closely related to the people and piggy versions. For the other 6 genes, the birdie virus formed distant monophyletic clades, which leads us to the thinking that the pandemic was not mostly caused by reassortment with sick bird genes. Where did the genes for our seasonal flu come from? The seasonal H1N1 virus contains PB1, PA, NP, and NA genes derived from the BM1918 strain via reassortment events that occurred while precursors to the seasonal H1N1 and the BM1918 strain co-circulated during the 1918 pandemic. The PB, HA, M, and NS genes arose independently from the BM1918 pandemic in approximately 1910, 1913, 1911, and 1915 respectively. As we fast forward nearly half a century, we can continue to trace the origin of the genes that played a role in the 1957 H2N2 pandemic. The H2N2 strain is comprised of 5 genes derived from the seasonal H1N1 strain, with the introduction of novel NA, PB1, and HA genes from reassortment events with Eurasian avian sources in 1950, 1954, and 1955 respectively. The introduction of new HA and NA genes may have led to this pandemic, as these glycoproteins are involved in the attachment of the virus to host cells, as previously discussed. Only a decade later, the H3N2 virus was made up of 6 out of 8 of the genes from the H2N2 strain, with the introduction of new HA and PB1 genes in approximately 1963 and 1967 respectively. As the H2N2 strain from the 1957 pandemic continued to circulate in human sources until last recorded in 1968, it is possible that the H2N2 and the H3N2 strains co-circulated for a short time leading up to the 1968 pandemic. Recalling that the genes shown in red were derived from the seasonal H1N1 strain, the blue from the BM1918 pandemic, and the green from the H2N2 pandemic, we can see how the genes were selected for, or against, with new iteration of virus to create a pandemic at its point in time. After reviewing various strains of the influenza virus, we have seen that the 3 most infectious strains were hybrid viruses with altered hemagglutinin subtypes. These viruses originated in swine, avian, and human hosts. The influenza virus has many strains, and only a small proportion have evolved to create pandemics.